Alrighty, good afternoon, everyone. So today's recording is going to be on mitosis and meiosis. So how I like to remember mitosis and meiosis are two different things. So meiosis will produce your gametes, your sex cells. So in men, sperm, in women, eggs. Talking about sex. Anyways, so if you produce sperm or you produce eggs, those are called your gametes. Now, our gametes are formed under the process of meiosis, and I like to think, okay, sex cells, sex phase, like O phase, meiosis. Mitosis is where you produce an identical daughter cell from the original mother cell. And so mitosis, I think, okay, well, if you have an extra toe, I don't. I have 5. That'd be cool if I had 6. The possibilities. Then you're making a repeat. So mitosis, meiosis, the two different specials. Now, what is the process of mitosis and meiosis, and how are they different? And I found a couple of resources that I will copy and paste for everyone. But I wanted to show you all of what I found. And so this website, technologynetworks.com/cell science/articles, mitosis versus meiosis, they have some cool clip art or pictures that I thought were helpful for students learning mitosis and meiosis. So the general process of mitosis and meiosis have similar steps: prophase, metaphase, anaphase, telophase. And if you go into molecular biology or cellular biology and microbiology, then you focus a little bit more on the phases. But in general biology, we say the 4 phases. And now, so each one has those phases and interphases. But the main difference, the takeaway, are the number of steps. Mitosis is a 4-stage total thing, while meiosis, you're going to repeat mitosis twice. So mitosis is a one-time thing. Meiosis, you got to do the phases again. And what I mean by that is right here. So each will go through prophase. That's just kind of getting everything set up, duplicating chromosomes, and whatnot from your parent cell. Metaphases are lining everything up. Anaphase is splitting up those chromosomes, and telophase is separating them out. Well, here we underwent meiosis 1. And that's just a fun way of saying mitosis 1, but meiosis 1. Mitosis, meiosis, they get confusing. Meiosis, all of meiosis is going to produce your gametes, and in a 2-part step. Mitosis is 2 identical daughter cells of the parent cell. And so again, you undergo prophase, so you set up your chromosomes, line them up in metaphase, split them apart in anaphase, and divide the cells, cleave on that telophase. And here is a really good drawing of that. So interphase is getting all that jumbly mumbly. Prophase, setting up your chromosomes. Metaphase, aligning them up. Anaphase, pulling them apart. Telophase, starting to separate them out and cutting them off. Those were our main 2 differences. Now, if we go back, The number of chromosomes in mitosis will remain the same, while the number of chromosomes in meiosis will be what we say haploid. So diploid, haploid. Well, what is diploid, you ask? Diploid, I'm glad you asked, is meaning 2N. We like to say 2 genes, so 2 chromosomes, and we say N. This is our diploid. Mitosis. And I think I spelled that wrong. Meiosis is 1N. This is haploid. These are the products of each one. They both have their prophase, metaphase, anaphase, telophase steps. Prophase, metaphase, anaphase, telophase. These are our biggest steps that we really care about. And mitosis, so you have your interphase. Mitosis will go through these steps once. One time. Well, meiosis will go through the steps 2 times. So if we were to draw this out and really draw the difference in the takeaway, let's say I have a parent cell with a jumble mumble of its nucleus and of DNA and chromosomes in its nucleus. Well, what it's going to do is it's going to unscramble its nucleus and start repeating, making copies of its own chromosomes. So remember, this is still 2N. And so, oops. Oh, I like this. Its nuclear membrane is breaking apart and it's aligning, it's making copies of its DNA, its chromosomes, breaking them up. And let's just say that those are supposed to be chromosomes. Okay. Also have we call these centromeres. Okay. So this is our prophase. Prophase. The next thing that we're going to do is that we're going to need to line all of this, all of our chromosomes up evenly, because remember, they're all copies. So originally that looks like a face. These are all copies. So I should do, we'll say the red is the copy of the blue. Okay, just so that we can see. So blue is the original, red is our copy. And what's going to happen is that all of our chromosomes are going to line up in the middle. So meta, middle, that's how I remember it. Meta, middle. And what's happening, oops, I want my blue. Is that these centromeres? Are attaching themselves to our chromosomes. So this is our metaphase. Cool. Metaphase, done. Our next phase is anaphase. So things are lining up in metaphase. Anaphase, we said, was pulling them apart, right? I know that I'm not being consistent with my drawings, but that's okay, because these are just examples. Make sure you try to be consistent. And I might have put too many of those little dots, whatever. All right, so they're being pulled apart. Oh no, but remember, this was still our original copies. So each one, this right here is still, so prophase, we've made our copies, so 2N. This, let's say, is 4N. 4N. This is still considered 1 unit, but it's being divided. So we, now that the chromosomes are separate, let's just keep it easy. Right now we're still in 4N. This is Anaphase. So anaphase, think Anna apart, A apart, Anna apart, Anna. That's how I remember it. So they're being pulled apart, the chromosomes. Our next phase, telophase. And telophase is going to be, we're going to start tearing our chromosomes. And that nuclear membrane is going to start forming again. And we're going to have a unique thing happening here. And it's called cleavage, where the cell starts to separate. But first it has to set itself up. So this is our telophase. And it's still together, so we would still say this is a 4N stage. After cleavage, so cleavage, meaning you're cleaving something, you're chopping it up into 2 new cells where genetic material is all scrambled up again. And they're identical. So 2 daughter cells. And this one is 2N, and this one is 2N. How did I get 2N from each one? Well, remember that this was 4N right there. So 4 divided by 2, because we split it in half, is 2N. And that's our general mitosis. Okay. Well, what about meiosis, you asked? Glad you asked. I do it sideways. That's okay. So we're going to start right here. Bam. We have our two daughter cells. Now, the one thing that makes meiosis unique is that we have something called the crossing over event, where genetic material might get swapped out with another piece of genetic material of the same chromosome, and that's what gives us variation, and it's random. Nothing says this piece is going to swap with this piece because it has to. It's all random, which is cool. Okay, so assuming that's already happened, we're going to undergo the two phases again. So in meiosis, I'm just going to copy what I did. Boop, 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 boop. This is the parent cell. Yes, 2N. Cool. Oops. We know that it's gonna duplicate its DNA. And color, let's use purple. So green, the original, purple. Okay. This is our prophase, right? Oh, and we should say this is meiosis 1. Still 2N. Oh, we copied our DNA, right? Should be 4N. Got to call me out. All right, prophase done. We know, well, prophase metaphase. So meta, middle. Our nuclear membrane has and each of these lines is supposed to represent 2 chromosomes, by the way. I'm just lazy and don't want to draw it as 4 different colors. Okay. So metaphase. Ooh, that's not metaphase, that's anaphase. I skipped a whole phase. Guys, call me out on this. Beep, beep, beep. We're lining things up. Nothing is separated. Metaphase, and this is still 4N. Cool. Alrighty. Now we can separate things. Now remember, this is still all meiosis 1. If you can recall, meiosis is 8 stages. Right now we're still in anaphase 1. This is still metaphase 1. Telophase, making our starting to do our little is a terrible partial circle. Material, genetic material, blah. Okay, telophase 1, done. Still 4N because we haven't broken up, remember. No, not technically 4N. The 2N, the 2N's are separating, but they're still together. I'm not gonna separate that because we care about the end product. All right, so after it's torn, we have 2 new daughter cells. Oh. That's our genetic material. Okay. We're not done because those clear membranes don't really stick around for long. Because what's going to happen is that it's going to go straight into our new phase of phase 2. Right. Looking at this. All right. And now, again, anaphase or metaphase, excuse me, is where we're gonna start pulling these ones are right. Shouldn't put a plus in. Yeah. Here. So before, you know how I did those copies, right? Each line I said was going to signify 2 chromosomes. So I'm going to keep consistent with that. Each line is still going to consist of 2 chromosomes. But now we don't have any duplication like we did in prophase 1. This should actually be in an interphase, I think. I'm going to have to double check this, but I believe this is called interphase. Double check that. I'll double check it and comment later. But let's focus on this. What happens with the next? This is Anaphase 2, or Metaphase 2, sorry. Why do I want to keep calling it Anaphase? Metaphase 2. Cool beans. It's still 2 of our 2n daughter cells. And now what's going to happen is that these daughter cells are going to pull little pieces of our original 2n cells. So remember that Punnett square explanation that I was talking about, and how our Punnett squares, we get one gene from one parent and one from the other? Well, it's all because of meiosis right here. See how each chromosome is being taken away? So now this is our anaphase. Or we still have 2 of our 2n, but now they're starting to be separated, right? They're being pulled apart, anaphase apart. And so ana apart leads us to telophase. So this is our telophase 2. Little bits of each genetic material. Okay. And now when we have this telophase, N, but remember that they're being pulled apart, and so yeah. All right, so now we have 1, 2, 3, 4, because right, 2 divided by 2 is still, or 2 times 2, because 2 divided by 2 is 1, but 2 of them each divided would be 4. So it becomes 2 divided by 1/2 is actually 2 times 2, which is 4. For those people who need to know the math. And we have 1. And each of these would be a gamete, new daughter cell. And if these ones right here each were 2, 2n, well now 2 divided by 2, that's 1. So that's just n. And we called right here at this beginning meiosis 2. Right? So this is meiosis 1 and then meiosis 2. So different end results than mitosis, 2 identical daughter cells, two daughter cells that are identical. And here we have 4 non-identical daughter cells. These are our gametes. Alrighty. So I'll just verify later the interphase is correct. But this is the general sense that we want to take away from mitosis and meiosis, is that with mitosis, that we're going to have a parent cell that is diploid that will undergo prophase, metaphase, anaphase, telophase. Pro meaning before, meta, middle, ana, apart, telo, tear. We can call it that, sure. That will produce 2 identical daughter cells. So mitosis to mitosis. Mito, so 2 sisters. My 2 sisters. Oh, mitosis. There, that's even better than mitosis. Mitosis. Oh wait. Mitosis. Mito. Yeah. And then meiosis, oh my goodness, I'm getting tongue-tied. You're going to have your parent cell, which will produce 4 non-identical daughter cells, and these are gametes. So meiophase, like meiophase, meiosis. So 4 gametes that are non-identical versus 2 identical daughter cells. Those are our main differences. And then there's more steps in meiosis than there are in mitosis. I mean, look at this drawing, like twice the amount, right? So that's what our main takeaway. I'm going to go ahead and post these little slide pictures that I found for your reference. If you have any questions, feel free to DM me, and I'll be happy to work with you.